so far we've been talking about typical short tandem repeats, the set of 20 that is used, but there's a couple of other types of DNA that can be used. So we'll talk about phenotyping, these YSTRs, and mitochondrial DNA. So one thing I can determine is that there is a particular gene called the amylogenin gene. It technically is one that's coding for tooth pulp, but it's different for by 6 bases in the X chromosome than in the Y chromosome. So it's found on the X and Y chromosomes, on the sex chromosomes, and that particular one will, because of that, will have 2 bands on the electrophoresis gel if it's from a male, and just 1 band because females have 2 X chromosomes. So it turns out by using that CODIS database, I can determine classical gender. So, in classical gender, you have XX and XY. There are some other types of gender that go in there, XXY, XYY, and that gets a little bit more complicated. But for traditional gender typing, you could determine that from the short tandem receipt, this typing that we've been talking about. So YSTRs is something else. So these are found on the Y chromosome. So in other words, you're only going to find it in men because only men have a Y chromosome. So for males, a YSTR repeat can be measured. And if that mixture of DNA shows many, like 3 different lengths, that suggests you can't get 3 different copies of that allele. You only have 1 per male because you get an X and a Y. If you're male, you'd have X and Y. So you only have 1 copy of that allele. And therefore, it can be used in cases of sexual assaults with multiple mates to be able to determine how many people were involved. And it can't really tell. You'd have to look at the rest of the typing, and if you had a mixture of DNA, that's pretty challenging, but at least can determine something about that there were multiple. Mitochondrial DNA is another kind of DNA. Remember, mitochondrial DNA doesn't have the full genetic code. So the nuclear DNA has the full, complete set of the genome with all the chromosomes in it. Mitochondrial DNA only has a portion of it. It's found outside the nucleus, which means we tend to find it more. It's a little bit easier to find mitochondrial DNA, but it will not be a complete set. And it is passed down from maternal lineage. So you can't tell a particular person. You can just tell like who they were related to in a maternal line. So in other words, you would have a mother and a daughter and a granddaughter would all have identical mitochondrial DNA. So you can't say which particular one it was, but it becomes kind of class evidence where you can say, yes, but there's a familial link. Again, but it only works with, the familial link will only work with women. So the last topic is phenotyping in this unit, is phenotyping. So genotype refers to what is your genetic code? CTG, ACC, so it's just what is coded. Phenotyping is saying, yes, but how does that code express itself in human traits? So now this is where we start talking about your green eyes or blue eyes, and brown hair or blonde hair, and your height, and all these things that get expressed by these different proteins that are coded. So, of course, at this point, we might be able to say that this DNA belongs to this person, or this person matches with this one, but we can't look at a genetic code and say, oh, this DNA happens to be somebody who has brown hair and green eyes. At least we haven't been able to do that. At this point now, scientists are beginning to understand enough of the code to start to say, I recognize that this person should have blue eyes, or this person should have green eyes. So this is a new emerging controversial subject method where basically you might be able to take somebody's DNA and try to come up with a profile of what they might look like, what their physical attributes might be, to kind of help with detective work. It's highly controversial. It's on the cutting edge. There's not a whole lot they can tell about it right now, but it is beginning. And also this is used for crimes in that sense. It's also used for genetic testing. There are certain cases where we know, okay, this particular area, if you have this code in your DNA sequence, you may be more likely to develop this kind of genetic trait. Certainly, breast cancers people are doing cell types to determine if they might have a particular higher risk of breast cancer or other kinds of cancers. So this is something that's sort of on the horizon. It'll probably be changing in the next 3 years, and everything I say today might be out of date. But just to know that this is something that's coming up in forensics.